Now, due to the importance of the production of genetically modified crops, we want to take a look at the latest progression of gene editing technology to see whether it is best. Let's take a look at how CRISPR compares to other gene editing technologies. There are two main factors to consider when looking at gene editing technologies. The first of which is the efficiency at which you get the required DNA modification. The second is off-target activity, which occurs when the DNA binding domains of the gene editing technologies bind to similar sequences to that of the target site. This can lead to the induction of double-stranded DNA breaks at unanticipated DNA sequences and the modification of genes which we did not want to happen. Now, there are a multitude of studies which compare CRISPR and other technologies in terms of efficiency and off-target activity. Here I have highlighted a few which would best be representative of a comparison between CRISPR and its other gene editing technologies. Here we compare CRISPR to TALENs. TALENs are a gene editing technology which have shown to have, particularly in plants, an extremely low induction of off-target activity, and as well as that, very high efficiency. Important to note that targeting a gene with CRISPR and TALENs requires each of the gene editing technologies to recognize a different sequence of DNA within that gene. The DNA binding domains of TALENs must first bind to a DNA base that is a thymine in a 3 to 5' direction. The specificity of DNA binding with TALENs can be quite high because the DNA binding domains can recognize sequences of up to 40 DNA bases. CRISPR can recognize DNA sequences of up to 20 base pairs. However, this sequence must be followed by a protospacer adjacent motif. Therefore, the sequences that we aim these technologies at inside a gene must differ. Scientists use Nicotina benthamiana plants to test the efficiency of CRISPR and TALENs. Here, each of these gene editing technologies targeted the luciferase gene in the plant. This gene, however, was purposely mutated with the addition of a DNA base in the middle of the DNA sequence. The gene editing technologies were targeted at this sequence, and once they bind to the DNA, they will remove this additional DNA base. Once the DNA base is removed, the gene will be expressed normally, and the scientists can then quantify the expression of this gene. This will represent the efficiency of the two technologies. Here we found that luciferase gene expression was approximately 11 times higher when using CRISPR compared to TALENs. This here shows that the efficiency of using CRISPR is 11 times higher than TALEN. Scientists also targeted the inositol phosphate kinase gene in maize plants. They used CRISPR and TALENs to target this gene and try to mutate it. They found that the efficiency of mutation using CRISPR was 13.1%, whereas using TALENs was 9.1%. Scientists also targeted the Wiskott-Aldrich syndrome gene and the tyrosine aminotransferase gene within jgwt2 T cells, which are essentially kidney cells. They looked at off-target activity within targeting these genes, and they found that using TALENs there was no off-target activity, meaning that the TALENs proteins did not bind to any other sequences other than the sequences in those genes. With CRISPR, however, the CRISPR machinery binded to 9 off-target sequences. The extent of the off-target activity was quite bad, where CRISPR would bind to DNA sequences that did not match that of the modified protospacer sequence. Ordinarily, CRISPR should bind to DNA sequences with a complementary sequence to that within the protospacer. It was found that CRISPR would bind to sequences that were not complementary. In such a way, it would bind to sequences which contained multiple DNA bases which were not complementary to the subsequent DNA bases within that of the protospacer. Some of the DNA sequences that CRISPR bound to had up to 13 non-matching DNA bases. The sequence in the protospacer should match that of the target DNA sequence. For example, in the diagram, the protospacer sequence of GCCG should match that on the target sequence of CGGC. But in this case, it seemed that the protospacer sequence was binding to DNA regions that were not complementary. On the right, we can see that the protospacer sequence of GCCG would bind to a non-complementary sequence, for example, CAAC. This is an example of the protospacer sequence matching a sequence with only two DNA bases which are not complementary. However, it was found that within the experiment that CRISPR would bind to sequences with 13 uncomplementary DNA bases.